Guess what time it is? Well, I guess we're a minute early, but that's okay because I'll just do some introduction and hey to everybody, especially those I can see, put a face to. Hey, Ayodeji. I'm sure I just killed that name, sorry. Hey, Hyatt.
Yeah, good morning.
How are you? Hey.
Hi, good morning.
Good morning. Good morning. It's midday here. You can see the sun is setting, so it's a little bit late. There's the great Drosophila right there.
On the brain back there.
And the brain. All right, let's get this party started. And I'm sure people will keep drifting in, and like I said, that's okay. So welcome everybody to day 2 of the Fabulous Fly Scientific Workshop, also known as the DrosAfrica Online Workshop 2020. And sadly, I remember we had this discussion. I wanted to tack on 2020 because I wouldn't be shocked if we had to go to also 2021. It'd be nice if we could do things in person, but we shall see. I am Ross Kagan, for those of you who don't know. I am a Regis Professor of Precision Medicine here at University of Glasgow, which I recently moved to. I'm a fly lab from way back, now studying cancer in flies, mostly drug development and clinical trials. We have an incredible lineup today. First, let me say thank you, everybody, for showing up and making this what I think is a pretty great workshop so far. Much appreciated. My thanks go out to our African science sisters and brothers. We're trying to create a community of elite fly workers, and your help with that has been much appreciated. And hopefully in return, we can bring you some knowledge on how you can use flies to drive your science forward in a practical way, but also an important way, in an impactful way. So, some Zoom rules. I am going to keep muting everybody. Sometimes people break through the mute. I just remute you. So if you're wondering why in the world you're remuted, I'm doing it. And that's to keep the general noise down, not to suppress discussion. On discussion, please put your questions in the chat session. If you would like to ask them, just make that note and I'll be happy to ask you to unmute and ask it when your turn. It's first come, first serve. The questions, incidentally, for those of you who weren't here yesterday, have been pretty amazing, and there have been a lot of them, which, respect for this. I really appreciate that. So of course we don't have time to ask all of those. So those questions have then been forwarded to your other questions on Slack. So for those of you who weren't here yesterday, we've opened a Slack channel, and it's a great place for everybody to continue the conversation after the talk. And in fact, I think, who was describing it? Oh, I think it was Sophia was describing it as Slack being on fire. And I think that's true. It was pretty rocking yesterday. And I think I learned a bunch of things just looking through Slack. So thank you for letting me steal your ideas. Today we have another poll. Okay, and the question is, have you ever worked with DrosAfrica? And I will give it a minute. So please vote. Have you ever worked with DrosAfrica? And that helps us know what kind of visibility that we have for those of you who signed in. I should probably be playing music now, but it's not going to happen. Hey, Mohammed, how you doing? Hey, Julia. Hey, Jose. Groovy. I think we have all our votes, or at least close to them. So I'm going to end this poll. So only about 28% of you have worked directly with DrosAfrica. I guess that's a fair number, but we need to make that, here, I'm going to share the results. We should definitely bring those numbers up. And now all of you who have been part of this workshop can now sign yes on that as well. For me personally, working with DrosAfrica, coming to Africa and meeting many of you has been really an incredible privilege. So it's been really good for me. A quick overview of yesterday. So we heard from, well, first we heard from Adam Dobson from University of Glasgow. He described how you can use flies to study how diet and in particular gut bacteria affect health and longevity. And we're going to hear more about that today, or at least longevity today. We heard from Amos, who gave a really beautiful talk on toxicology and using flies. And Amos really talked about some of the larger issues that are in play and how Drosophila can address those. Then we heard from Nicole Broderick from Johns Hopkins on fly microbial effects, microbiota effects, and not just Drosophila, but also tsetse flies, which I thought was really interesting, bouncing back and forth. And finally, we heard from Carlos Ribera, whose work, of course, was amazing, looking at the feeding decisions, but I think what was even cooler was his videos and images and stuff, which were pretty spectacular, and that was worth the price of admission right there. So that was a great first day. That will, of course, be overshadowed by our speakers today, who will do an even more impressive job, I'm sure. I want to, today's Nobel Prize winners, Fly Nobel Prize winners of the day are these folks right here. So a quick background for this 1995 Nobel Prize. So Drosophila was really begun as a model system back at the turn of the last century. First papers were published in 1904, 1905, and the discovery of the white gene. Drosophila was used in the early part of the 20th century as really the primary tool to work out the basics of genetics. And we talked a little bit about that yesterday and how that led to a pair of Nobel Prizes. And the genetics was really a formalism at that point. And during that time, the other great push was in embryology, sort of cut and paste biology. And the two finally merged with Drosophila workers such as Don Poulsen and in particular, Ed Lewis, who you see there. So Ed really carried the baton of fly genetics as he worked out the basic body plan and the factors, homeobox proteins that drove that body plan. And I wouldn't say he worked alone, but it was to some extent in isolation. There were other important workers, Seymour Benzer and so on, who were doing key work. But Drosophila was really not a primary model system for a lot of that time. And then sort of a convergence of two important threads. One, molecular biology techniques were just being worked out, and some of those in Drosophila by Dave Hogness, by Jerry Rubin, by Alan Spradling when they worked out P elements. Hogness worked out basic cloning from mutation to gene. So all this molecular biology stuff was just being worked out at the same time that these two folks, Eric Wieschaus and Janni Hussein-Volhart, did this groundbreaking screen, one of the most important experiments in Drosophila, where they basically did a saturation screen for all the genes required in the zygote, zygotically, for a young embryo to be assembled. And I remember at the time when this was going on, and Janni Hussein-Volhart in particular was given a hard time because the estimate in the number of genes required to make a young embryo was estimated to be roughly 1 million to millions. So the actual number of zygotic genes required, at least the ones they identified, was 32. And the total number of genes, as it turns out, in a fly genome is about 15,000. In us, it's roughly 24,000. So the millions of genes turned out to be not correct. And that identifying those key genes, they identified hundreds and hundreds of genes, but those central genes and having the ability to clone them and identify what the sequence was happening at the same time in the 1990s, those two worked together to make Drosophila really the driving force in the genetic basis of development, and then really worked out a lot of the basics of how the young embryonic plan is laid down using Drosophila. It was really a pretty heady time. At the same time, signal transduction became big. I was part of that, as were many others, and the field in general got much larger. And that's really laid the foundation for modern Drosophila research, for modern Drosophila work. And these three were rewarded for driving developmental genetics forward with a Nobel Prize in 1995. And in fact, Eric Wieschaus was sort of my de facto PhD advisor back at Princeton. And good guy, I have to say, all very interesting people. So, my last point on that is, so our last Nobel Prize was 2 years ago, circadian rhythms, so I'll probably mention tomorrow. But of course, we're going to need more Nobel Prizes, and Africa seems like a perfect new incubator for new fly Nobel Prizes. And so I look forward to continuing to ask a lot of key questions, just encouraging this. And today, our speakers, which I'm really excited about, is the topic is disease and aging in Drosophila. We're going to begin with Hamed Badmos, who I met, I don't know, I guess many months ago. He invited me to give a talk, and now I'm pretty excited because at some point he's going to come to the lab, which is really great. He is really a brilliant young man doing beautiful work. And I thought also if he could talk a little bit about what it was like as an African native coming to the UK, and what it was like to be a scientist, and as well as show off some of his science. Then an old friend and somebody that I have admired for many years, Tano Gonzalez, who's really one of the pioneers in using Drosophila for cancer. And so getting him to come here and speak today, I'm pretty excited about this. And then, importantly, learning from yesterday, we're going to take a 15-minute break, okay? And we're going to give you a chance to stretch, chance to grab some food. So yesterday's session probably could have used a break in it, and especially we're going to have an additional talk today. So after 15 minutes, we're then going to move to Laura Towert, who's somebody that I have enormous respect for, who is an entrepreneur who has reached across to use fly technology in a company, My Personal Therapeutics. And I think this is really going to be an unusual and enlightening fly talk, which how you can use Drosophila to drive a company forward. And I'm actually really looking forward to this as well. I've had a lot of interaction with Laura. Then, as I mentioned earlier, Linda Partridge, who is one of the absolute giants in the aging field, also uses Drosophila and has been on quite the roll lately, which is impressive because she's been on a roll for a while. And she's going to talk about how using flies, not just to study aging, but actually finding solutions to extending lifespan. And finally, and this is special for those of you who are trainees, but I think everybody, including myself, can benefit from listening to Mohammed Salem from Tantayu in Egypt. And he's going to talk about the role of the Center of Excellence in Research in fostering biomedical research in Africa. So that's a lot. And so I think it's time that we get started. So Hamid, are you ready to share your screen?
Yeah,
Excellent.
Let's go for it.
Perfect.
Yeah, the file is a bit massive, so might be a bit slow. Let's see. Good. So are we good to go?
Go for it.
Good. Thank you very much for the invitation, Ross and the DrosAfrica team. So my name is Ahmed. So I'm presently rounding up my PhD in Bennett Lab at the University of Liverpool. So I'm going to tell you some story about the Drosophila egg. So I think you've heard a bit from Carlos yesterday about the fantastic things you can do when it comes to inter-organ communication. But I'm only going to focus on this particular organ and the cell types in that organ. so a bit of background about myself. So I started uni at the age of 16 at the University of Ilorin in Nigeria. So, and then moved to the UK to finish university degrees and then finishing up my PhD now. So, and presently I work in one of these institutes, and it's quite a beautiful area and a cool serenity to work in. And I hope you guys can visit one of these days. And we're surrounded by cool people and cool guys that are always ready to help. And I think my science has been really channeled by people that I'm surrounded by, because as an undergrad, you need lecturers, you need people that can support your academics. And then as a very young person, you need people that you can look up to and be like, oh, I want to be like them. So, and moving to the UK can be a bit of a challenge, but once you're surrounded by the good people, by the kind people that can boost the potential in you, then you can strive and you can do whatever you want. So I'm very lucky to be part of both University of Ilorin and also University of Liverpool. And luckily, I've spent a lot of my life in the university building, and I'm very excited to continue to do that. So, and hopefully moving very close to Liverpool, hopefully also moving to Glasgow soon to continue my research in Roskegan's lab. So also, I'm part of the pioneers of DrosAfrica Nigeria, and all thanks to Rashida Abdulaziz for putting this together, because she has done a fantastic job, and you can see her pictures there. And I think most of you might have met her in the past. So she's a very good leader, and she put us together to do this type of job and like giving back to the country and then making sure that the generation experience what we're unable to experience. And because DrosAfrica is a fantastic model, and you can do cool stuff with them, and you don't really need millions of funds to do that, but like you can do everything with tens of thousands of naira. So thanks to Rashida for pioneering this, and you guys can get involved, and you can. So I think the DrosAfrica Nigeria is focused in the north at the minute, so you guys can take to the southwest, to the southeast, and also to different parts of Africa. So today I'm going to tell you about the Drosophila egg. So the Drosophila egg is a very good model for both cell migration and epithelial integrity. And I'm going to tell you about this complex known as the Saga complex and the proteins that are components of this complex. Also, I'm going to tell you some story about how these processes are very important for both cancer and tumorigenesis. So I'm gonna be honest that Drosophila don't develop cancer, but I think some studies from Kaitana's lab recently shows that maybe Drosophila can develop cancer. And also you can, so it's gonna tell you some cool stuff about the real cancer stuff that I'm not gonna really touch that. So, but I'm gonna tell you about what really the phenotypes you might be looking for when you're studying the Drosophila egg. And I think at the end of the day, you're gonna appreciate what we can do with the cool egg of the fly. So the big question here is, how does cell acquire the ability to migrate, and how does cell break the barrier for them to migrate? And this transition is what is called the epithelia to mesenchymal transition. So it is the ability of cells that are localized or located at the epithelium. They might proliferate, they might not, but they tend to break that barrier and then start migrating. So because I've got loads of videos, I'm only going to use my arrows here. So they can then migrate. So it is of note that this is very important for both development and in disease, especially in cancer. And you can and what the leading killer of cancer is cancer metastasis. Not only can cancer migrate from the site from the primary site through the blood barrier to secondary site, they can migrate both collectively and individually. Also, they tend to interact with the microenvironment, especially the immune cells. And these immune cells can either enhance their migration or inhibit their migration. So this is a very complex system, and it's a very complex process in human, but you need some models to study this. So one of the models that people use, and researchers, we want something easy, something simple, something you can understand, something you can play with. So one thing is the C. elegans, so you can study the breakage or the disruption of the basement membrane in C. elegans easily. Also, you can study what is that signal. So what is the chemo attractant that signal to cell? So why will cell want to migrate from here to here? So what is that signal that signals to them? So you can study that in amoeba. So amoeba tends to look for food via a gradient of chemo attractant. So you can study the simple mechanism of chemo attraction in amoeba. Also, you can go to vertebrate like zebrafish, and you can study, you can generate transgenic zebrafish, and you can begin to study cancer cells, and you can begin to study cancer metastasis in this organism. And these are not very expensive, and they're very simple. And my favorite of all, and I think the most powerful of all, and I think if it is not that powerful, I'm not going to be here, is a Drosophila egg, or Drosophila as an organism. And I think most people have shown you the power of why we used Drosophila organism yesterday, and why because they're really cheap, because they've got a short life cycle, because we've got genes that are conserved from Drosophila to human. So, but I'm going to tell you about a process of migration known as the border cell migration. And also, I'm going to tell you the formation of the epithelium, and also when there was abnormal epithelium, what that leads to. So I'm going to really focus on Drosophila egg. So in Drosophila, there was a pair of ovaries, and the ovaries are made up of ovarioles. So, and the building block of the ovarioles are the eggs. So the eggs develop from the germarium, which you've seen yesterday from callus stalk, the houses, the stem cells, and they're required for the production of adult eggs until matured eggs are laid and fertilized. So there are two types of cells in these Drosophila egg. So the germ cells, which are the bigger part, so the germ cells, the nest cell, or the oocyte, and the oocyte, and also the somatic cell that surround the germ cells. Like I mentioned, Drosophila has been, so the egg has been very useful in studying in stem cell research. Also, you can study cell proliferation and how cells differentiate. So why would cells choose a germ cell fate instead of a somatic cell fate? So you can begin to study the genes that are required for these. Also, importantly, you can study cell migration, which is what I'm going to focus on today, especially border cell migration. You can study the maintenance of cell polarity, and also the formation of the epithelium in the eggs. And also you can study cell death. So these are things you can study with the Drosophila egg, and because you can easily image them. So as a cell biologist, I'm very interested in believing what I can see. So I can tell, is there a problem with the epithelium? Are the cells dying? What is really going on with this with the organ that I'm studying? So I can easily see everything. And that's the power of microscopy. So also you can study genes that are required in development and disease. So you can begin to ask, okay, the egg developed from the germarium to stage 6. What are the genes that is going to stop that? What are the genes that is going to enhance that? So instead of 15 nerve cell, why are we going to have 30 nerve cell? Instead of 1 oocyte, why are we going to have 2 or no oocyte? So you can begin to ask those type of questions. And especially you can begin to look at disease. You can begin to look at genes that are involved in aging, in various neuronal disease, in cancer especially, and also in regeneration. So these are the things, these are the power, these are the things you can use the Drosophila egg for as a tool. And also, like, there is a workshop for DrosAfrica, so you can begin to think, okay, what are diseases that are peculiar or that are relevant to Africa? What are those things that we can study in these flies, and then you can appreciate those things, and those, you can begin to understand the mechanism of those things in fly. So not only that, you can also do drug screening. So you can do toxicity screening. So you can begin to look at, okay, what are those things that is going to prevent egg from laying? What are those drugs that are going to enhance egg laying? So, and that also comes back to reproduction. And so the power of Drosophila egg for both cell and developmental biology, as I mentioned, is that we can image them. And not only can we image fixed sample, we can also image them in real time, like so what we call live cell imaging. So, and also you can begin to quantify these imaging. So you can do simple quantification of the morphology, of the characteristics of the activity of the cells. Also, you can do a sophisticated analysis of these cells. So also you can do an analysis of the whole organ or the whole tissue as a whole. So you can begin to ask what, so you can see that the egg tends to rotate. So there are some genes that are required for this rotation. So you can begin to ask how long, what is the speed of rotation? How long does it take for them to rotate? So you can begin to ask that question. And you can begin to ask what are the genes that are required for these, not only in migration, but also the rotation of the egg, because all these are very important for the egg development. So that is a bit of intro into Drosophila eggs. So I'm going to tell you a bit of the technology that we routinely use in the lab. So Drosophila genetics has got a wealth of tools, and honestly, it's just easy for a young scientist like myself to like dive into the system and utilize the wealth of knowledge that are available. So you might have heard of the GAL4 US system from talks from yesterday. So I'm going to tell you a bit about Markham technology that was developed in 1999. So there are some references at the back that can refer you to that. And I can always send you papers about this. So what Markham does is it allows you to generate labeled cell in an otherwise unlabeled background. So what that means is, assuming let's say you've got a mutant of interest. Let's say you want to study a protein and you generated a mutant of that protein, and you want to really understand what is the role of that protein in a particular system, let's say in cell migration, in cancer. So what you can then do is you can tag that protein or the mutant allele of that protein with what we call FRT, so flippage recognition target. And then you can, the reason why you need to do that is because most of these mutant allele are homozygous lethal. So you need to make sure that you can also balance them. You will combine them to what we call FRT. And then you then cross that particular flight to what we call a tester strain. So, and the tester strain has got loads of genetic elements, including GAL4, including your UAS that can allow you to label. And also particularly it has got flippase that can flip out the FRT site. So flippase is an enzyme that does that trick. So, and then what you do is you cross these flies together and then you generate some offsprings, and then the offsprings has got loads of elements. And then what you then do is you can shock these flies at 37 degrees specifically, or a bit lower, but not higher because they're gonna die. So, and then that induces mitotic recombination. And then after cell division, you can begin to label cells either with GFP or absence of GFP. So you can tell, and not just GFP, you can label with LAC-Z, you can label with RFA. So, but we use GFP routinely because that's easy for us to see, and also because of the type of microscope that we use, and also because ideally in Drosophila world, people tend to tag things with GFP. So it's just easy for us to do, and that's just a way to go. But you can tag with new, so there are new fluorescence protein now, like BFP, so blue fluorescence protein, you can tag with that as well. So, but the idea here is just for you to label your cell, and then it is of interest that these cells are they're mutant for your protein of interest. So, and cells that are lacking GFP or the fluorescence protein are wild type basically. So this is a particular tool that we use routinely in the lab, and most of the data that I'm going to show you today, they were generated using these methodology or using this technology. So part one of my talk is going to focus on cell migration, and you can read more about these what I'm going to talk today, what I'm going to tell you about today in this preprint that we've just published. And there's a link, and you can easily scan there, and that's going to take you to the paper as well. And I'm happy to send you a PDF if you want. So what we found is that non-stop is also known as Drosophila USP22. I'm going to tell you detail about that later. Is required for border cell migration. Also, it regulates F-actin polarity, and also it is required for the EPO signaling. I'm going to tell you about that. And also the role is independent of what we'll call the Saga at module. So also independent of the whole Saga complex. So in the lab, we're very much interested in cell migration because cell migration is required for both cellular activity and in disease, especially cancer, right? And that is the basics, the basis for cancer migration or cancer metastasis. So in the lab, we're utilizing a model known as the border cell. So border cells are cells that migrate from stage 9 to stage 10 of egg development. So they're specified as stage 8. You can see that they are the epithelium. After specification, they undergo what we call EMT, epithelium mesenchymal transition, and then migrate towards the oocyte. So I'd love to tell you that there are two types of cell in this cluster. So the polar cell that is required for the specification, and also the outer border cell that coordinate the collective migration. So this is a form of collective migration, not single cell migration. And the video below shows you a 4 hours time lapse of how these cells are migrating. So normally the cell migrate between 4 to 5 hours. So it depends on who you ask. So, but these are 4 hours border cell migrating, and the migration is dependent on, so they need to be properly specified, they need to be properly timed. So migration can never start at stage 10. So if you don't see migration at stage 10, it's never going to happen, because the cues or the signalings that are required for that migration are upregulated at this stage, and then that enhances the migration. So if you fail to see anything at stage 10, then they're not going to migrate. So that's a failure of cell to migrate. So the timing needs to be properly tackled and needs to be properly understood. So also, they require normal actin cytoskeleton. So what is actin cytoskeleton? Actin cytoskeleton is one of the key factor, or one of the key tool for cell migration. So if you ask anyone in the cell migration field, they're going to tell you about, oh yeah, we need actin, or we need to check for actin. So it is required. So it is one of the key protein that is really required for cell migration. And as you can see in this video, the cell tend to elongate at the beginning, and that is just the protrusion that is generated via actin cytoskeleton. So actin is really required for them to migrate. So if they fail to generate that protrusion, then they're going to fail to migrate. And I'm going to tell you that there are two phases in this cell migration. The running phase, where we've got a leader cell that leads the cell migrating, and also a rotating phase at this end, where cells tend to rotate and then migrate collectively. So those are the two phases in migration. Also, the cells that are migrating needs to be properly adhered together. So, and people have shown that this is really regulated by polar cell, by proteins that are localized in polar cell, echidherin, armadillo, and all these. So these are the cell-to-cell adhesion needs to be properly maintained. And also the number of cells that migrate needs to be properly specified. So there are about 6 to 8 cells that are migrating. Anything below or above that leads to abrogated migration or failure of cell to migrate. Also in the lab, we're very much interested in a family of protein known as the ubiquitinases or DOPs. So some labs are interested in kinases, some labs are interested in phosphatases, some labs are interested in E3 ligases, but we are interested in DOPs. And what DOPs are enzymes that remove ubiquitin from proteins that are targeted for degradation. And also what they do is they allow for the maintenance of free ubiquitin in the system, because any abnormality in the homeostasis of free ubiquitin leads to various neurodegenerative diseases, including Parkinson's. And these are enzymes that are required to maintain that level in the system. So there are about 95 dobs in human, and there are 45 dobs in Drosophila. So in a screen for the 45 dobs in Drosophila, we find non-stop as a gene required for border cell migration. As seen in the control, cell migrate normally towards the border of the oocyte at stage 10, but when we knock down, so when we reduce the level of non-stop in border cell cluster, they fail to migrate. So I've seen, this is the quantification here. So it is well mentioned that non-stop is part of a complex known as the Saga complex. So the Saga complex is a co-activator of transcription, and it is required for both acetylation and deubiquitination of histone 2A2B. So the acetylation is catalyzed or monitored by the histone acetyltransferase module and the several components of this module. And also the DOB activity of the complex is also monitored by what we call the DOB module. And one of the components is non-stop. And we've got ataxin 7, which is like an allosteric regulator that regulates the stability of the whole complex. So I'm gonna come back to ataxin 7 in a bit. So non-stop is also known as USP22, but in human, so it is USP22. And the name USP22 is from ubiquitin specific protease. So those are the names that are given to mammalian or human protein. But in Drosophila, non-stop came from the name that so non-stop came from the name that doing axon targeting in neuron. So neuron needs to stop at the lamella of the photoreceptor, but non-stop mutant failed to stop. So that was why the name non-stop. But in border cell, they stop. So assuming we discovered this first, probably we might have named it stop protein. So that is the gist about our names. Our come up with name in Drosophila. So to further understand the role of non-stop in border cell migration, so we generated Markham clone, and then we begin to watch the characteristics of this clone. So in control, so in wild type control, you can tell that the cell continues to migrate with a single protrusion towards the oocyte, not labeled here. So the migrate normally, and you can see the track of the control. But in non-stop mutant clone, you tend to see tumbling. So the cell failed to migrate, but they continue to tumble, as seen from the track here as well. So using a software that we routinely use in the lab, so we segmented this cluster into front extension, side extension, and back extension. And then we begin to characterize the biasness of this extension for both the control and the absence of non-stop. So we found that in control, there was a bias for front polarity or front extension in the cluster, but in non-stop mutant clone, we find bias for side extension. And this logically tells us that the reason for the side extension induced the tumbling, and that is the reason why cell failed to migrate. So then we want to understand what are the effectors, what are the pathways that are disrupted in these clones. So previous study have shown that actin cytoskeleton is localized at the cortex of this cluster. So these are about 6-7 cells, but the actin staining is always at the cortex of those cells. And you can draw a line across the cluster, and you can measure the level of actin in the cortex and in the junction. And we can tell that the actin cytoskeleton is biased for the cortex. But in non-stop mutant clone, we found that there was a rush, or there was accumulation of actin into the junction of the cells of the cluster, and then you can begin to quantify that, and that was what we saw with the quantification as well. Previous study have shown that the EPO components, especially a study from Barry Thompson lab and Nick Tapon lab at the Creek in London, have shown that EPO components are localized at the junction to prevent the accumulation of actin into the cortex. And this is required for normal border cell migration. So what is the EPO pathway? So EPO pathway is a pathway that is conserved from Drosophila to human, and it is required for both proliferation and growth. And also it has been, the components of this pathway is implicated in various cancer types. And in border cell, it is required for normal actin polarization and also normal polar cell proliferation. So these are the components of the pathways, and the components are localized at the junction of border cell. So we picked two components, the chromes, so chromes that is required for the stability of other components, and expanded, which is also required for stability and also combined to chromes. And it has been shown in the past that this expanded can be a target for non-stop. So we ask if this can be targeted in non-stop mutant clones. So can non-stop act via the EPO pathway? So indeed, Crohn's is localized at the junction, and that was what we saw with our quantification as well. But in non-stop mutant clone, we saw redistribution of Crohn's into the cortex. So, and clearly this experiment shows that non-stop is required for normal localization of Crohn's, which restricts actin to the cortex. But in the absence of non-stop, then Crohn's is mislocalized to the cortex, and then that allows for actin to accumulate into the junction. So clearly, non-stop is acting upstream of junctional hypocomponents. So also we ask if non-stop can regulate the expression of the IP components. So we looked at expanded LAG-Z. So in cell biology, you can instead of you doing like PCR or qPCR, there are some tricks that allows you to measure expression of genes in your own in any context that you're interested in, especially in cells. So we can measure the expression of expanded using expanded LAG-Z. So this wild type control, the wild type labeled with GFP, or also wild type in the absence of GFP, you can see that the level of expanded is similar here. But in non-stop mutant clones, so non-stop, in the absence of non-stop labeled with GFP, there's a reduction in the expanded. And also, so in the wild type, wild type that has got non-stop but absence of GFP, there's an increase in expanded. And that's a quantification there. So clearly, this experiment shows that non-stop is required for the expression of components of EPO pathway, and probably reduction of this expression allow them to redistribute to the cortex by binding to something that we're still trying to figure out at the minute. So then the next question is, we ask was that, okay, can we put back expanded, and can that rescue the phenotype of non-stop? So, and these are things you can do with the Markham technology as well, which can be difficult with other technology. So then we ask, what would be the phenotype of increase in level of expanded? Is too much expanded bad for beta cell cluster? So we find that expanded on its own is all like the clusters are happy with expanded increase, and Crohn's localization is normal because it is at the junction. But it is of interest that we can also rescue the phenotype of non-stop with increase in expanded. So you can see with this in this image that Crohn's is now properly localized to the junction. And here is the quantification. So in control, Crohn's is at the junction. In non-stop mutant clone, it is at the cortex, not at the junction. Increase in expanded is normal, and also expanded can also rescue the phenotype that we saw in non-stop mutant. So the next question we ask is, okay, is non-stop acting on its own or via the Saga complex? So we picked two components of this complex. So ADA2B, which is a component of the histone acetyltransferase model, and non-stop, and ataxin 7, which is required for the stability of the complex. And we begin to ask, can the phenotype of this phenocopy the phenotype of non-stop? So we found that ataxin 7 is required for border cell migration as seen in this quantification, and also for actin polarity. But ADA2B, which is a component of the histone acetyltransferase model, is not required for both border cell migration and also actin polarity. So this experiment clearly shows that the role of non-stop is independent of the ADS model. It is of interest that while 6 to 8 cells migrate during border cell migration, we saw as much as 30 cells in ataxin clone, and I really love it. This is a rare phenotype to find. And this is a phenotype that we also saw with non-stop, and that has been reported for the EPO pathway. So in concluding, ataxin 7 and non-stop are required for border cell migration independent of other components of the Saga complex, especially the ART module. And this is required for the expression of genes that power of the EPO signaling, and then that allows them to localize the junction and restrict actin to the cortex. But in the absence of this complex, there's a redistribution in the EPO components, and that leads to accumulation of actin into the cortex, into the junction, and that leads to tumbling and failure of cell to migrate. So at the moment, we are asking if the catalytic activity of non-stop is required because it's a DOB, right? So also if this is dependent on histone 2A, 2B, and if these mechanisms are conserved in other Drosophila tissue, because we can begin to look at immoside migration and begin to ask what is the role of non-stop in this type of migration, and also we can begin to look at the brain disk and and the eye disk to see if that leads to any tumor phenotype. And it is of interest that USP22 has been shown to be overexpressed in some cancer. So we can begin to ask, are the effectors that we found in border cell migration the same effectors of USP22 in cancer? And that can allow us to better drug these cancers. And it is of interest that dogs generally are difficult to drug. So if you can find a bad effector, like something downstream of them, and if you can drug that instead, then you are good to go. So that is the end of the first part, and this is the last part of my talk. So we're looking at we're looking at ataxin 7 and epithelial integrity. So we found that ataxin 7 is required for normal epithelial architecture, and also this affects notch activity, and this is dependent on Saga activity. So the egg epithelium is made up of what we call the somatic cells, and this is required for both egg development and can be on onset for tumorigenesis. So I would love to mention that the epithelium divide the egg into both the apical layer in yellow and also the basal layer. And you can begin to study what are the characteristics of this layer and how can you disrupt them. So what is ataxin 7? Ataxin 7 is a component of the Saga complex. So germline mutation in ataxin 7 has been implicated in various neurodegenerative disease. Somatic mutation in this gene can cooperate with RAS in thyroid cancer, and it has been shown recently that it can also regulate actin cytoskeleton. So then we generate MACAM clone in the Drosophila epithelium and ask what is the phenotype of loss of ataxin 7. So in control clones, there was a single layer of epithelium, and that is required for organogenesis or egg development. But in ataxin 7 clone, we saw multi-layering of this epithelium, and also in some cases you tend to see them invading into the oocyte. So this clearly shows that you can model an aspect of tumorigenesis in Drosophila, especially in the egg. And you can begin to ask what are the genes to add, what are the genes to remove, what are the drugs to add to prevent this type of phenotype. So one thing that we ask is that, okay, what are the targets? So there are two phases in egg development. The mitotic phase that regulates proliferation phase, and that stops at stage 6, and the endocycle phase where cells stop proliferating but increase in size and rearrange to increase the size of the egg. So, and one of the regulators of this is the notch pathway. So expression of notch immediately after stage 6 prevents proliferation. So we ask, so the excessive cell that we saw at the posterior end, is it due to loss of notch activity? So one of the downstream effects of notch is COT, to monitor the activity of notch. So indeed, at the beginning, you tend to see COT expression, and because notch is activated, COT is lost at the late stage. But in ataxin clone, COT is there at the beginning and also there at the end. And this is specific mostly to the anterior and posterior end of the egg, and also specific to where you've got GFP positive cells that are labeling the absence of ataxin 7. So clearly this shows that notch activity is affected in ataxin 7 clone. So the next thing we ask is, okay, ataxin 7 is required for the stability of the complex, right? So is this acting on its own, or is it acting via the catalytic subunits of this complex? So we knock down non-stop in the posterior end, and we found that notch activity is normal. We looked at other 2B clone, and we also found that notch activity is normal. But when you combine these knockdown and loss of function mutation together, then that leads to loss of notch activity. So this clearly shows that the activity of the complex is required for ataxin 7 role at the posterior end of the egg. So in concluding, I've shown you that there's a requirement for normal epithelium, and that is required for normal organ development. So in the absence of some protein, especially components of the Saga complex, that leads to multilayering and loss of notch activity due to the expression of COT and cyclins. I did not show you data for polarity, so we looked at polarity. We look at so there's a polarization for both the organ, the anterior posterior end, and also polarity in the somatic cell. So we look at these, and we found that both of them are affected in ataxin clone. And it is of interest that ataxin 7 has been implicated in thyroid cancer. So you can begin to develop thyroid cancer model in flies. You can begin to then drug that, or you can begin to do genetic screening with that. So far so good. I've shown you the power of Drosophila egg to model both cell migration and epithelial integrity. And as a proof of principle, I've shown you the role of some genes in this process. And it is worth mentioning that these two processes are required for both cancer and tumorigenesis. And also this study shows us that different population of cells respond to different gene perturbation. And you can begin to interrogate all the genes, if you like, and begin to understand them. So if you're interested in what I've shown you so far, these are some labs that work routinely on the Drosophila egg. And so the Alan Spradin labs, one of the pioneer of the field, and Dennis Monter, pioneer of the border cell migration. So St. Justin in Cambridge, one of the pioneer of the polarity. So you can begin. So you can talk to people, you can meet people, and you can ask them questions about what they've done. And I'm happy to share these later on. So, and to fellow young scientists like myself, I'm going to tell you that don't be afraid to try new things, because so if you've got an idea or a better way to do things, try and find good people to support you, because with good people and great ideas, it always seems impossible until it's done. So thank you very much for listening, guys, and I'm open to any question.
Hamid, that was brilliant.
Thank you.
I always love to see a young, brilliant scientist on one of their early talks when you're there. So remember this, people. You have way too many questions, so I'm going to ask you some of them, and then you're going to have to go to Slack to get the rest. So thanks, everybody, for these questions. And actually, I'm going to, ridiculously, I'm going to start very quickly, because you mentioned several times finding a good mentor. Any advice on how to do that?
Very good question. I think, honestly, so the best people that you can talk to at the minute are probably your supervisors or your lecturers, but look beyond your universities, or you look beyond your institute. Like, people are always out there that are ready to up. So you don't really need to look, assuming you're in University of Ibadan, you don't need to look just in Ibadan. You can look for people in Unilag, you can look for people in Egypt, you can, so like tread beyond your physical environment. So like you can ask from people around, and the world is a university on its own, and is like the Twitter is there. You can meet people on Twitter. Like, people are interested in your ideas. So once you've got good ideas, people are ready to talk to you. But make sure the approach is okay as well, because first impression goes a long way. So make sure the impression that you're giving people is the right one and not the wrong one. So I think that's one way.
Yeah, no, I think that's very thoughtful. I would agree with that. All right, let's go to the science. So Miriam Adeniki Saladin, asks, how similar is the Drosophila egg to the human eggs or follicles, particularly in terms of gene composition?
Very good question. So yeah, so there's a lot of similarity between the Drosophila egg and the mammalian and the human egg, but also there are a lot of differences, right? So one thing is that the human egg is more sophisticated or is more complicated compared to Drosophila egg. Drosophila egg is very simple, right? Just few thousands of cells. So, but the human egg is a lot more cells, right? And also, so most of these genes are conserved. So, like the genes that are required for laying egg in human are also required for laying egg in Drosophila, but there can be some layers. So not necessarily. So you can pick a small layer in Drosophila, and you need to go a bit higher when you're looking at human eggs. So there are a lot of similarity and differences. Yeah.
Sure. Falmi Smo wanted to know, he says, great talk. Actually, that's pretty much, there's about 30 great talks there. So if you want to feel good about yourself, read those. I wanted to know how you generated the real-time images. Is it by confocal microscopy?
Yeah. So you need a very good hand. So like Carlos said, so when you're dissecting the border cells, so because they can so the mechanics of the tools that you're using will affect their migration. So you have to be very careful when you're dissecting them. So you need a good and you need a good microscope. So we use just simple confocal microscope that has got temperature control and humidity control as well. So yeah, simple.
Okay, cool. Mohammed Jameh wants to know if you have any info about the positioning of the nucleus during the migration. Does this change if we manipulate the centrosome key proteins?
Very good, very good point. So yeah, there are people that have looked at that. So you can read paper from Dennis Montel's lab. So she has looked at proteins that are located in centrosome, and definitely these will affect border cell migration. But it is worth mentioning that border cells are not dividing while they're migrating. So these are post-mitotic migration, and it is of interest that these are in the endocycle phase. So they don't migrate. They also so I was talking to her recently, and she was like, the only thing that happened in border cell is that at the end, they tend to have just a single round of DNA replication. So nothing happens between that migration to the end. The only thing that happens is that cell need to adhere properly. They want to migrate, so they've got single polarity, the number needs to be coordinated. But when it comes to the nucleus, there's nothing really going on in the nucleus. If something is affected at the beginning, then they're not going to migrate. So, and that is where the nucleus is very important.
Sure, cool. So De Kaiser wants to know what is it? It's such an interesting question. What is at stage 10 that prevents the cells from migrating? Because can you use that to actually fight cancer metastasis?
Good. So you mean at stage 10 they don't migrate? Is that what you're saying? So good. So at stage good. So upregulation of some gene is required for them to migrate. So if they don't migrate at stage 10, is either one reviewer was like, why don't you check cell death? So probably these cells are they're dying. So that was why they don't migrate. Is that okay? And also assuming they want to migrate, but they fail to migrate, that's another thing. So some of them might stand still, not doing anything. So that might probably because they're dead. While some might still be proliferating, which then we can use that as a cancer model, right? Then we can begin to study that. But if they don't migrate at stage 10, then they won't migrate. Then we can nitpick what is really going on, what are those pathways that they're required, and what are those things that is not there. So then we can begin to look at that.
Yeah, I know, De Kaiser, that's an interesting question. I hadn't really thought about that. And Gero Mohammed asks, how maternal effect genes control early Drosophila development, and I think in particular, how do they impact creation and migration of those border cells? The maternal effect genes. That's a
yeah, that's a long one because these are adult flies. So I think when you're talking about maternal effect, so maternal effect is gone at the early stage of embryo, and if something like so what can happen is I might have forgotten to tell you that if border cell fail to migrate, then there is nothing like egg development. So, and also because so like study from Dennis Montella have shown that so in the slow mutant, the sterile, they don't fertilize it. So you won't develop into embryo, but maternal effect at the embryo stage, it doesn't have any effect with borders of migration in adults. Yeah. So.
You're gonna have to draw all the genes out from the mother.
Yeah.
Two more quick questions, and then we'll move forward. Lola asked, does nonstop also regulate other membrane-associated proteins such as FAST3, FASTCON3, or is it specific for transmembrane proteins downstream of Hippo? So have you just focused on Hippo because that's what you care about, or does it regulate other things?
I think I focused on Hippo because of the time limit of the talk, right?
Right.
So we've shown that FAST3 is affected actually, and also because this cell tends to split. So one of the phenotypes that I did not show here is that the cell splitting in non-stop mutant. So FAST3 is affected, and also not only FAST3 but also armadillo, disc large. So these are things that are located in lateral and apical domain. So these are affected as well. So but the time limit is there, right? So but yeah, so you can so the paper we've shown everything in the paper and
okay, well, all I can say is don't write a check you can't cash because they're going to get you in Slack when you hear about it.
No
last one, and then we'll move forward. I'm from Nahir Ghazal. Really nice talk. We want to know, are ataxin 7 mutant eggs viable?
Very, very brilliant question. So actually, we've not checked that routinely, but ataxin 7 destroys the egg. So definitely they're not viable. Just tell, like, even though you lay them, they're not going to fertilize. So it destroys the egg. So I don't think they are. Yeah. But we've not really checked that carefully.
Okay, cool. Again, Mohammed, many thanks. That was fantastic. Really enjoyed that.